Good morning students. Good morning everyone. In this video tutorial, we are going to learn about intermediate reaction, and Krebs cycle. In the last video, we have discussed about glycolysis. It is the first step of respiration. In the last video, we have discussed about glycolysis. It is the initial step of respiration. Respiration has 4 sequence of steps, that is glycolysis, intermediate reaction, Krebs cycle and electron transport chain. In glycolysis, what is the product of glycolysis, where it occurs? Glycolysis occurs in cytoplasm. The product of glycolysis is 2 molecules of pyruvic acid, net molecules, that is 2 molecules of ATP, 2 molecules of NADH. The fate of glycolysis is formation of pyruvic acid. This pyruvic acid has 3 fates. After formation of pyruvic acid, if the oxygen is available, it enters into the mitochondria and continues with the intermediate reaction. If the oxygen is available, pyruvic acid enters the mitochondria and continues the intermediate reaction and Krebs cycle electron transport chain. If oxygen is unavailable, that is anaerobic conditions, in aerobic conditions it undergoes intermediate reaction and Krebs cycle. In anaerobic conditions, that is oxygen is unavailable, it may undergoes fermentation in some bacteria to form ethanol, or in some cells like muscle cells it forms lactic acid. In muscle cells it forms lactic acid. So pyruvic acid has 3 fates. One is whether in the presence of oxygen it may enter into the mitochondria and continues with the intermediate reaction, or in absence of oxygen it undergoes fermentation in some bacteria and form ethanol, and in some muscle cells where oxygen is unavailable it forms lactic acid. First we go with the intermediate reaction. Before going to the intermediate reaction, this pyruvic acid is formed from glucose, which is formed from carbohydrates. This pyruvic acid like molecules is also derived from proteins and lipids also. Lipids and proteins also produce pyruvic acid like molecule and they undergo Krebs cycle reaction and they form energy in the form of ATP. Not only glucose, from carbohydrates, proteins and lipids also undergoes Krebs cycle reaction by formation of pyruvic acid like molecules and forms lots of amount of energy in the form of ATP. First we will discuss about intermediate reaction. What is intermediate reaction, where it occurs? Intermediate reaction or pyruvate oxidation or linked reaction. Another name is linked reaction. Pyruvate oxidation or intermediate reaction or linked reactions are same. It occurs in mitochondria. Whenever the oxygen is available to the pyruvic acid, it undergoes pyruvate oxidation in the mitochondria. First, we will discuss about one by one these points. If the oxygen is available, aerobic respiration will proceed. If the oxygen is unavailable, anaerobic respiration will proceed and forms lactic acid. In some bacteria, alcohol is also produced by the process of fermentation. In eukaryotic cells, pyruvate molecules produced at the end of glycolysis are transported into the mitochondria. These pyruvate molecules are entered into the mitochondria, especially matrix, mitochondrial matrix. Once inside the mitochondria, one carbon dioxide will be removed from the pyruvate, leaving an acetyl group that will be picked up by the activated by the carrier compound called coenzyme A. Removal of carbon dioxide from any compound is called decarboxylation. One carbon dioxide molecule is removed from the pyruvic acid and forms acetic acid or leaving this acetyl group, and this acetyl group is picked up by the coenzyme A. The resulting compound is called acetyl coenzyme A. Coenzyme A is made from vitamin B5, that is pantothenic acid. This acetic acid formed by the decarboxylation is added to the coenzyme A, which is vitamin B5 pantothenic acid, and forms acetyl coenzyme A. During the formation of acetyl coenzyme A from pyruvate, 2 electrons are also transferred to NAD+. Removal of hydrogen ions or electrons from any compound is called oxidation. Hence the name pyruvate oxidation. Here pyruvate acid undergoes oxidation by removing 2 electrons or 2 hydrogens from its molecule, hence it is known as pyruvate oxidation. And also one carbon dioxide molecule is also removed, hence it is called decarboxylation. Hence it is a both oxidative decarboxylation reaction. Together we call as oxidative decarboxylation reaction. Two electrons are transferred to NAD, nicotinamide adenine dinucleotide, and forms NADH. Pyruvate dehydrogenase is a multi-enzyme complex. It contains 3 sub-enzymes. that is pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. Enzyme 1, pyruvate dehydrogenase, which uses thiamine pyrophosphate as its cofactor. Dihydrolipoyl transacetylase, which uses lipomide as its cofactor. These are all cofactors of these enzymes. Dihydrolipoyl dehydrogenase, which uses FAD, flavin adenine dinucleotide, as its cofactor. This pyruvic acid undergoes decarboxylation, removal of one carbon dioxide molecule and removal of two electrons or two hydrogens, and adding these electrons or hydrogens to NAD plus and forms NADH oxido and forms acetic acid, and with this acetic acid is added to the coenzyme A, which is B5 pantothenic acid, and forms acetyl coenzyme A, and this acetyl coenzyme A enters the Krebs cycle. What are the exact reactions of these three enzymes we will discuss here. Reactions of PDH complex, pyruvate dehydrogenase complex reactions. Pyruvate dehydrogenase with TPP, thiamine pyrophosphate as coenzyme, decarboxylates pyruvate to hydroxyethyl thiamine pyrophosphate. What pyruvate dehydrogenase does? Pyruvate dehydrogenase with the help of TPP decarboxylate pyruvate. This pyruvic acid is decarboxylated by pyruvate dehydrogenase and forms hydroxyethyl thiamine pyrophosphate. Then dihydrolipoyl transacetylase catalyzes the conversion of hydroxyethyl thiamine pyrophosphate into acetyl lipomide and then transfers acetyl group to coenzyme A to produce acetyl coenzyme A. Once again, dihydrolipoyl transacetylase catalyzes conversion of hydroxyl ethylene pyrophosphate, what forms here hydroxyethyl thiamine pyrophosphate, into acetyl lipomide and then transfers acetyl group to coenzyme A and produces acetyl coenzyme A. And the last enzyme, third enzyme, dihydrolipoyl dehydrogenase catalyzes conversion of reduced lipomide into oxidized lipomide by transferring the reduced equivalence of FAD, and it completes the cycle. Now this acetyl coenzyme A enters the Krebs cycle. Now we will discuss the Krebs cycle. Krebs cycle. It is named after the scientist Sir Hans Krebs. Sir Hans Krebs. It is also called as citric acid cycle. Citric acid cycle. And another name is also there, that is tricarboxylic acid cycle. Tricarboxylic acid cycle, that is TCA cycle. It has three names: citric acid cycle. The first formed product is citric acid, hence named as citric acid cycle, and it was discovered by the scientist Sir Hans Krebs, hence known as Krebs cycle. And it is also called as tricarboxylic acid cycle, TCA cycle, because citric acid contains 3 carboxylic acid groups. We will discuss it later. 3 carboxylic acid groups, hence it is also known as tricarboxylic acid cycle. We will discuss the first reaction, that is condensation of acetyl coenzyme A with oxaloacetate. Acetyl coenzyme A formed in the linked reaction or intermediate reaction combines with oxaloacetic acid in the presence of enzyme citrate synthase and also involves one molecule of water and forms citric acid or citrate. Here, acetyl coMgMa is a 2-carbon compound and oxaloacetate is a 4-carbon compound. Both combine to form 6-carbon compound citric acid. The oxaloacetate reacts with acetyl group of acetyl coMgMa. This is the acetyl group. CH3CO is the acetyl group. Acetyl group of acetyl coMgMa and water, resulting in the formation of 6-carbon compound, that is citric acid and coMgMa. The reaction is catalyzed by the enzyme citrate synthase that condenses the methyl group of acetyl coMgMa. This is the methyl group. CH3 is the methyl group. Condenses the methyl group of acetyl coMgMa and carbonyl group of oxaloacetate. This is carbonyl group. Carbonyl group of acetyl coMgMa, resulting in citryl coMgMa, which is later cleaved to form free coMgMa and citrate. Once again, acetyl coMgMa from intermediate reaction reacts with oxaloacetate. This is 2-carbon compound, this is 4-carbon compound in the presence of water molecule, and also citrate synthetase enzyme forms 6-carbon compound, that is citric acid. This is the condensation of acetyl coMgMa with the oxaloacetate. And the next step is isomerization of the citrate into isocitrate. This step involves two sub-steps. That is, now for the further metabolism, citrate is converted into isocitrate through the formation of intermediate cis-aconitase. Citric acid is converted into cis-aconitic acid. First, citric acid is converted into cis-aconitic acid by losing one water molecule by dehydration in the presence of the enzyme aconitase. And also in the next step, again by rehydration, by adding another molecule of water in the presence of the same enzyme aconitase, here aconitase and here also aconitase, forms isocitrate. Here citric acid and isocitric acid has the same molecular formula but different structures. Here we see, here it contains, this carbon contains OH group and this carbon contains H group. Here the position of the H and OH is different. Hence they are known as isomers. These two reactions occurs in the presence of the enzyme aconitase. This reaction takes place by the two-step process where the first step involves dehydration, removal of water molecule from citrate and forms this aconitic acid, followed by the second step involving rehydration, again adding of another molecule of water to this aconitic acid to form isocitrate. And third step is oxidative decarboxylation of isocitrate. This is also a two-step process. Oxidative decarboxylation of isocitrate. As we discussed in the pyruvate dehydrogenation, here also oxidation occurs, decarboxylation occurs. Removal of one carbon dioxide molecule and adding of 2 electrons from the substrate to the NAD occurs here. This is the first oxidation reaction of the Krebs cycle. There are 4 oxidation reactions occurs in this Krebs cycle. This is the first reaction. The third step of citric acid cycle is the first of 4 oxidation reduction reactions in this cycle. Isocitrate is oxidatively decarboxylated to form 5-carbon compound alpha ketoglutarate. Isocitrate is a 5-carbon compound. Alpha ketoglutarate is isocitrate is a 6-carbon compound. Alpha ketoglutarate is a 5-carbon compound. Isocitrate removes one carbon dioxide molecule from its structure and also 2 electrons and 2 hydrogens, and it gives to the NAD plus, forms NADH. This is catalyzed by the enzyme isocitrate dehydrogenase. Wherever dehydrogenase comes, NAD and the NAD molecule gets reduced to form NADH molecule. There is exception to this reaction we will discuss here. This reaction, like second reaction, is a 2-step reaction. In the first step, isocitrate is dehydrogenated to oxalosuccinate. This dehydrogenated to form oxalosuccinate. This is a 6-carbon compound. And the second step involves decarboxylation of oxalosuccinate to form alpha ketoglutarate. Decarboxylation to form alpha ketoglutarate. Both the reactions are irreversible steps. These are all irreversible steps catalyzed by the same enzyme isocitrate dehydrogenase. The first step, however, results in the formation of NADH, while the second step involves the release of carbon dioxide. Here, isocitrate is first dehydrogenated to form NADH molecule and forms oxalosuccinate. This is a 6-carbon compound. This oxalosuccinate undergoes decarboxylation and forms 5-carbon compound alpha ketoglutarate and thus forms oxidative decarboxylation reaction. This is the first oxidative reductive reaction in Krebs cycle. Next step, oxidative decarboxylation of alpha ketoglutarate. This step is another one of the oxidation reduction reactions. This is the second oxidation reaction in Krebs cycle. Alpha ketoglutarate is oxidatively decarboxylated to form 4-carbon compound succinyl coenzyme A. Alpha ketoglutarate is a 5-carbon compound. It undergoes oxidative decarboxylation reaction to form 4-carbon compound, that is succinyl coenzyme A, and 1 molecule of carbon dioxide, and also forms 1 molecule of NADH. The reaction is irreversible and catalyzed by the enzyme alpha ketoglutarate dehydrogenase. Here again dehydrogenase. Wherever dehydrogenase comes, NADH molecule forms. Found in mitochondrial space. This reaction is similar to oxidative decarboxylation of pyruvate involving reduction of NAD plus to NADH. Alpha ketoglutarate undergoes oxidative decarboxylation and forms succinyl coenzyme A and carbon dioxide, and 1 molecule of NADH also forms here. Next step is 5th step, conversion of succinyl coenzyme A into succinate or succinic acid. This is the next step. Succinyl coenzyme A undergoes an energy conserving reaction because GTP molecule is formed here, in which succinyl coenzyme A is cleaved to form succinic acid or succinate. Succinyl coenzyme A is cleaved to form succinic acid and coenzyme A. This occurs in the presence of the enzyme succinyl coenzyme A synthetase. This reaction is accompanied by the phosphorylation of guanosine diphosphate. Phosphate group is attached to GDP and forms GTP here. This GTP is immediately, the phosphate group present in GTP molecule is immediately given to ADP and forms ATP later on, depending on the type of cell where it is present. And this is known as substrate level phosphorylation. The reaction is catalyzed by the enzyme succinyl coenzyme A synthetase. Here succinyl coenzyme A is converted to succinic acid. Step 6. Dehydration of succinate to fumarate. Succinic acid to fumaric acid. Dehydration. The name itself indicates removal of water molecule from the substrate. Dehydration. Here succinate formed from succinyl coenzyme A is dehydrogenated to fumarate, catalyzed by the enzyme complex succinate dehydrogenase. Found in intramitochondrial space or inner membrane space. Inner mitochondrial membrane. This succinate dehydrogenase is present in inner mitochondrial membrane. Inner mitochondrial membrane. This is the only dehydrogenation step. This is the third oxidation step. Third oxidative reductive step. This is the only dehydrogenation reaction in citric acid cycle in which NAD plus doesn't participate, where FAD participate. Instead of another high energy electron carrier, flavin adenine dinucleotide acts as a hydrogen acceptor, resulting in the formation of FADH2. The FADH2 then enters the electron transport chain via complex 2, transferring the electrons to ubiquinone finally to form 2-ATP. This reaction will discuss in electron transport chain. Succinic acid undergo dehydrogenation reaction and forms fumaric acid and FADH2 molecule. Instead of NADH, FADH2 molecule is formed. It occurs in the presence of the enzyme succinate dehydrogenase. This is the special kind of enzyme which is present in inner mitochondrial membrane. Hydration of fumarate to malate. The fumarate is reversibly hydrated to L-malate in the presence of the enzyme fumarate hydratase or fumarase. As it is reversible reaction, formation of L-malate involves the hydration, whereas the formation of fumarate involves the dehydration. Fumarate undergoes hydration to form L-malate or malic acid. It undergoes in the presence of the enzyme fumarase. 8th step, the last step, dehydrogenation of the L-malate to oxaloacetate. This is the 4th oxidoreductive step. 4th oxidoreductive step. The last step of citric acid cycle is also an oxidation reduction reaction where L-malate or malic acid is dehydrogenated to oxaloacetic acid in the presence of the enzyme L-malate dehydrogenase. Malic acid dehydrogenase, which is present in the mitochondrial matrix. This is the reversible reaction involving oxidation of L-malate and reduction of NAD plus into NADH. 2 electrons and hydrogens are removed from the malic acid and transfers its electrons to NAD plus, forms NADH. And forms oxaloacetic acid. This is a 4-carbon compound. This is also a 4-carbon compound. This oxaloacetic acid, as we discussed earlier, where we start the Krebs cycle, that is from oxaloacetic acid, is regenerated here. This forms the complete cycle. Oxaloacetic acid thus formed allows the repetition of the cycle, and NADH formed participates in the oxidative phosphorylation reaction, that is electron transport chain. This reaction completes the cycle. Once we summarize what we have discussed, acetyl coenzyme A formed in the intermediate reaction enters the Krebs cycle by condensation of oxaloacetic acid. This is 4-carbon compound, and acetyl coenzyme A 2-carbon compound forms 6-carbon compound, that is citric acid. In the presence of the enzyme citrate synthase, this citric acid undergoes dehydration rehydration reactions. In the presence of the enzyme aconitase, the intermediate compound is cis-aconitic acid. This cis-aconitic acid is converted to isocitric acid. This is also a 6-carbon compound. Isocitric acid is converted to alpha-ketoglutarate acid by first oxidoreductive reaction, dehydrogenation reaction. This isocitric acid is converted to alpha-ketoglutarate acid. Alpha-ketoglutarate acid is a 5-carbon compound. Isocitric first undergoes decarboxylation reaction and then dehydrogenation reaction. This adds its electrons and hydrogens to NAD plus, and forms NADH. This alpha-ketoglutarate acid undergoes another oxidative reaction, forms succinyl coenzyme A. This is the second oxidoreductive reaction. Here alpha-ketoglutarate is a 5-carbon compound. Succinyl coenzyme A is 4-carbon compound. One molecule of carbon dioxide is removed, and NADH also is produced in this reaction. This succinic acid undergoes substrate level phosphorylation and forms GTP. This GTP gives its phosphate group immediately to ADP to forms ATP, substrate level phosphorylation. Succinyl coenzyme A undergoes cleavage to form succinic acid and coenzyme A. It undergoes in the presence of enzyme succinyl coenzyme A synthetase. This succinic acid undergoes another oxidation, that is third oxidation, and it forms FADH2. It gives its electrons and hydrogens to FAD plus, not NADH, and forms FADH2. It undergoes in the presence of the enzyme succinate dehydrogenase. This is the special enzyme which is present in inner mitochondrial membrane. This succinic acid undergoes hydration. Succinic acid undergoes hydration to form fumaric acid. Dehydrogenation undergoes fumaric acid. This fumaric acid undergoes hydration to form L-malate or malic acid. This malic acid undergoes fourth oxidation, oxidoreductive reaction. One NADH molecule is formed here. Malic acid in the presence of malate dehydrogenase and forms oxaloacetate, where we have started is regenerated here. This is oxaloacetic acid, and it continues with another round of Krebs cycle. Here, as we observe here, acetyl coenzyme A contains 2 carbon atoms, which is used up in the form of by releasing 2 carbon dioxide molecules in Krebs cycle. Pyruvic acid releases its first carbon dioxide molecule in intermediate reaction, where second and third carbon dioxide molecules are removed in Krebs cycle. Thus completes the Krebs cycle reaction. What are the byproducts of the Krebs cycle? We will discuss here. What are the key products of Krebs cycle? ATP. The cycle directly produces one molecule of ATP or GTP per turn for each acetyl coenzyme A, as pyruvic acid produces 2 acetyl coenzyme A. 2 pyruvic acid are produced, 2 acetyl coenzyme A are produced, so 2 ATP. For each acetyl coenzyme A, 1 ATP, 3 NADH, 1 FADH2, 2 carbon dioxide molecules, 1 ATP is produced. For 2 acetyl coenzyme A molecules, 2 ATP, 6 NADH, 2 FADH2, 4 CO2 molecules are released. This is the key products of the Krebs cycle. Thank you for watching this video. Subscribe to this video for more videos like this.